I'm Maria-Grazia Spillantini. I am Professor of Molecular Neurology here at the University of Cambridge, but I am Italian. I come from Tuscany and I study biology at the University of Florence. I was born in Caprese Michelangelo, near Arezzo. We are in what is now called Clifford Allbutt building, but actually until 10 years ago it was the Laboratory of Molecular Biology. The thing is that when I was in Italy, I was working on migraine and on pain. And Cambridge, that is also the reason why I end up in Paris. I'm working with Jean-Charles Schwartz on encephalitase. And then I came to Cambridge because here there was Iversen working on an enzyme that was involved in degrading substance P that at that time was believed to be part of the pain system, involved in pain. But then I started the PhD here in Cambridge with Aaron Klug and working with Michel Godet, who was a senior postdoc at that time. When Michel identified tau, we worked together and we identified the 6 tau isoform in human brain. We cloned them and we find that these 6 tau isoforms, they are all involved or aggregated in Alzheimer's disease, and they become hyperphosphorylated. And that to say was a sort of a bit of a serendipity that brought me to this field, but then it has completely caught my attention, my passion, and we start to look at families that they had, for example, forms of fronto-temporal dementia. At that time, although they had been identified mutation in the APP gene that were causing Alzheimer's disease, the tau protein was believed not to be relevant because there were no mutation leading to Alzheimer's disease or any neurodegenerative disease. However, I was involved in looking at brain to do immunohistochemistry, to look at the neuropathology of cases with neurodegeneration. And with Bernardino Ghetti, we start to study a family from Texas that had a great tau pathology. And so I characterized the pathology in this family and also in another family from Seattle. Some people did not find tau in the brain of their patients, and so because of my expertise on tau staining and tau pathology, then at that point many people sent me brains from their patients, and it came up that actually all these cases except one, they had tau pathology. The reason why it had not been seen was because routine methods were used for the staining, like silver staining or so on. And instead, I used immunohistochemistry. So I was able to see tangles, even if they were not very, very tightly, so insoluble. And therefore, all this family clearly showed tau pathology. And so in the family from Texas, I found strangely that different from Alzheimer's disease or also from PSP, progressive supranuclear palsy, that we had previously seen, there were only tau with 4 repeats forming the filaments. There was no tau with 3 repeats. So we look at the tau gene, we sequence it, and what we found was a mutation in the tau gene, which was difficult to see because it was in the intron, was not in the cDNA in the normal expressed sequence. But this mutation in the intron downstream of exon 10 was regulating the splicing of tau isoform. In particular, in tau, the 6 isoform differ for the presence of 3 or 4 tandem repeats in the C-terminal part of the protein, and this mutation increased the spliced in of exon 10, increased the amount of tau with 4 repeats. And this was important because in human brain, the ratio between tau with 3 and 4 repeats has been reported to be 1. So it indicates that just a change in this ratio actually led to pathology and led to tau aggregation, and we still don't know why. But so we found one of the first mutation in the tau gene, and this was it's called +3 mutation. And because of the amount of tau pathology that we found, we call this family multiple system tauopathy. And this was the first time the term tauopathy was used. So this was a very important finding, and then other two groups identified mutation in the tau gene exactly at the same time. So these three papers that identified the normal missense mutation, these three papers were published in June 1998. Over time, then we start to study inflammation in the brain of patients with Alzheimer's disease, and also with fronto-temporal dementia. Instead, we use one of the mutations that we had identified, the Pr and this was in a family coming from northern of Italy, from Genova. And this family, the father developed the disease at the age of 29 and the son at the age of 28. So we thought that in a mouse that lives only for 2 years, we need something aggressive, and therefore we use this transgene that now is used in many other transgenic mice. And so these mice develop tau aggregates, tau pathology, within 7 months, which is quite quick. But this is also good to understand the mechanisms. And so we found that in these mice there was a lot of microglia activation, and over time we have continued to study this sort of inflammation. And now we have more recently found that the microglia in these mice becomes abnormal. So initially the microglia doesn't react to the tau expressed only in neurons, but then when you start to have the aggregates and the filament, the microglia phagocytes the neurons containing tau, and after that, it's like it gets indigestion, becomes senescent and does not work anymore, and start to spread the tau that it has ingested, leading to the spreading of the pathology and the disease. In our mice, there are specific areas of the cortex where neurons die, and so we try to transplant neuronal precursor cells in the brain of the mice in order to replace the neurons that had been missing. What we found instead is that these cells, they had become, instead of a neuron, astrocytes, but they were able to rescue the neuronal death that we had observed. So why these astrocytes were able to do so when actually there was astrocytosis in the brain of the mice that was accompanying the tau pathology? We became curious why the endogenous astrocytes cannot rescue neuronal death. And so we start to study in vivo in the mice, but also in vitro, in primary culture of astrocytes, what was happening when neurons got tau. As I said, tau was expressed only in neurons, but we could see that the astrocyte already, mice few weeks old, they were dysfunctional, and therefore they were unable to rescue the neuronal death. They were not supporting neurons anymore. Extending these studies, we found that there were proteins not expressed and released by the astrocyte, like for example thrombospondin, who are involved in are neurotrophic and neurosupportive. And so when we add thrombospondin to the media, we could rescue the deficit found in the astrocytes. And finally, more recently, studying more the astrocytes, we found that actually something that goes wrong is that the astrocytes have this abnormal release of proteins that normally are in the cytoplasm, but we found them in the media where they are contacting also, they are released, and then the media is toxic for the neurons. Now we are trying to understand how this is happening and if this is happening in a human brain. In all these studies, what we have done, we have investigated and confirmed the finding in the mouse brain, in a human brain from Alzheimer or for patients with frontotemporal dementia. The other thing is that while we were working on tau and studying tau modifications, we identified two proteins that were not known, and one of our antibody was recognizing the antibody for tau recognized them. So we cloned these proteins and we named them alpha synuclein and beta synuclein. We made the chromosomal localization of the two proteins in chromosomal 4q21 for alpha synuclein and 5 for beta synuclein. And so when Parkinson's disease was hereditary form of Parkinson were linked to a locus on chromosome 4q21, we knew that synuclein was there. During our study, we had developed antibody for the protein. We have many reagents. And so what we did, when then actually the genetic mutation was localized in the alpha-synuclein gene, we obtained Parkinson brain from the brain bank, but also some dementia of Lewy body tissue slices from John Trujanowski. And then I stained all of this tissue, and we found that indeed alpha-synuclein was in Lewy body. And not only we did not have any genetic form of the disease from the family with the mutation, but these were sporadic cases. And so this was the first identification of alpha-synuclein in the Lewy body. Then we characterized the filaments from Lewy body, but also from multiple system atrophy gliocytoplasmic inclusions, and we found that they contain alpha-synuclein. And so now these diseases are known as alpha-synucleinopathies. Now we know, according to Brack, that the disease Parkinson could start based on the presence of Lewy body in the gut and then moving to the brain, or from the nose and moving to the brain. So now in the lab at present we have three main projects. One is producing organoids made by human iPS cells, and these organoids we are using to study the tau pathology, and the organoids contain astrocytes besides neuron. We are trying to determine if they are able to form neurofibrillary tangles or tau aggregates, and then in collaboration with Michel Godert and chercheurs, we will do cryo-EM to see if the filaments that they form have the same structure of filaments extracted from the brain of Alzheimer's disease patients. The other project is to work again on microglia to understand better the dysfunction that occurs after ingesting tau, and in particular the astrocytes. We are comparing the characteristic of the astrocyte derived from human iPS-induced pluripotent stem cells with those that we obtain from mouse brain primary culture, and then determine if there is the same dysfunction, and we have preliminary data indicating that this is the case. For what concerns synuclein, we have transgenic mice that express the protein in dopaminergic neuron of the substantia nigra. We express an aggregation-prone form of the protein, and we are testing compounds in collaboration with various groups. For example, one is ANLE138B that now is in phase 2 clinical trial. So our results show that it could work. It works in mice, rescue the dopaminergic deficit, both prevent neuronal death. And then now we are studying another small molecule, treating the mice and study all these aspects. But also we have mice that express synuclein in the gut or in the nose, and we are studying the mechanism of spreading from gut and nose to the brain, and try to understand the mechanism in order to identify targets for therapy. And this is what is going on in the lab at the moment. And I am very grateful to the foundation for the prize, because it has been really important for myself. That was a tremendous ceremony that we had in Paris, the honor of receiving the Paris, but also for my group, because with the grant, with the prize that we obtained, it has been possible to support the research on astrocytes. And we have now published our first paper on the mechanism of dysfunction of the astrocyte that also hints to the human astrocyte, and we acknowledge the foundation because contributed partly to the salary of some persons involved and also to the consumable that we have used. And it is really an honor for me to have been awarded this very important prize.